Thank you so much. So as was mentioned, my name is Shammamah Hossain, and today I'm going to be talking to you a bit about Bio. So just a little bit about myself. I graduated from McGill University with a joint degree in physics and computer science, and I've been working as a software engineer at Plotly ever since June of 2018. So I'd like to get to know you guys a little bit more. So just by a show of hands, who has used Dash or Plotly within the past, let's say, couple of months? Okay, good. And another show of hands, how many people here are bioinformaticians? Great, awesome. So hopefully I'm not going to bore anyone today. So first I'm going to give you guys an introduction to Dash and bioinformatics for those of you in the audience who might not know what either of those are. Next up, I'm going to go into some of the motivations for why we created this library. I'm going to walk you through a specific subset of the Dash bio components that I personally find the most interesting. And then I'm going to take a little detour to talk about an auxiliary library that was developed during the development process for Dash Bio. Then I'm going to wrap it up with some demonstrations that show some typical use cases. So Dash Bio is a Plotly product, but what is Plotly? So some of you might have heard of us. Like I said in my first question, we have a really popular graphing library, plotly.py. But this is only one part of Plotly, the company. So Plotly as a company offers another product called Dash, which is super important, and that forms the entire context of what we're going to be talking about today. The best way to describe Dash is that it's a tool for building analytics applications. And what I mean by that is any application that a data scientist would use to better visualize or understand their data. Dash runs on a Python or an R backend, and Dash applications are web applications, so they run in your browser. The Dash family contains several component libraries. So we have Dash core components, and that contains things like dropdowns, sliders, and text inputs that the user can use to interact with the application. We also have Dash HTML components. So those are used to define the layout of your app, which I'm going to show you in just a second. We also have a couple of specialized libraries. So we have Dash DAC for data acquisition and control. So that would be used in something like a manufacturing process and contains components like gauges, knobs, and indicator lights. And of course, Dash Bio, which I'm talking to you about right now. And the most important thing about Dash is that everything is MIT licensed, open source, and free. So everything that I'm about to show you today, you can easily go home after the conference and run all of this code yourself and be able to replicate what I'm going to show you. So to create a Dash app, it's really simple. All you have to do is open up your favorite text editor, which should be Emacs, by the way. And then you have to define a layout for your application, and then you have to define the logic or the behavior of your application. So up here, I've defined the layout as just a container, and then immediately underneath it, a text input box. And then the behavior of the application down here, I've defined as whenever the user enters a value into the text input, we perform some operation on it. So in this case, that would be capitalizing all of the letters. And then we return the result of that operation and display it above. So this is exactly what you see here. And you'll see that also it is running in my browser. It's currently running on localhost, but you can imagine that since this is a web app, you're able to serve these to many users if you want. So maybe this is not exactly the application that you'd want to show off to people because it's quite simple. It's not very exciting, but we do have a huge community of users who are very excited about Dash. And one of those is Max Shen, who was a bioinformatician. So this application that you see on the right was actually developed to be a supplement to a publication that was published in Nature. So as I'm sure all of you know, research papers and research topics are incredibly specific. So a tool like this could not be purchased off the shelf. This is not software that exists. It is software that was created specifically for this purpose. And it was created, as he mentions, in only a handful of days. The other cool thing about this is that, like I mentioned, since Dash apps are web applications, not only do you not need to download any software, but you also don't even need to be on a computer to visit and use this application. You can visit it on your mobile device and it will work exactly the same way. So So let's talk about the other half of the equation, which is bioinformatics. Bioinformatics is a subset of data science. So where data science combines statistics, computer science, and some domain-specific knowledge. In the case of bioinformatics, that particular domain is biology. Bioinformatics is typically employed in fields like medical research, in clinical trial analysis, genomics, and proteomics. So why would we choose to marry together DASH and bioinformatics? Well, we've seen a rapid growth in the field of bioinformatics, but more importantly than that, we've seen a rapid growth in the use of the phrase big data in bioinformatics papers. So that to us signals a need that the bioinformatics community has for easy-to-use analytics tools just like DASH that can integrate into their existing workflows. And why is DASH perfect for bioinformaticians? Well, like I mentioned, DASH contains component libraries, and those component libraries essentially are sets of Lego blocks that you can put together in virtually infinite permutations to create an application that is very specific to your needs. And we know that by the existence of libraries like Genome Diagram, BioPython, and GeoParse, that bioinformaticians are already using Python for their data analysis needs. So DASH would integrate seamlessly into their existing workflows. Okay, so we're going to have another little interactive bit here. Could you raise your hand if you know what any of these charts are, or if you've seen something like them before? Great. Okay, so these are all of the DASH bio components. There's quite a lot, so in the interest of time and your attention, I'm going to just focus on a few of them. We have three main types of components. So we have three-dimensional rendering tools, we have custom chart types, and we have sequence analysis tools. Our first 3D rendering tool is SPEC. So this is a beautiful WebGL-based molecule renderer. You're able to play with the molecule by zooming in on it, moving it around, and rotating it. And it uses techniques like ambient occlusion and depth of field in order to give the user a much better idea of the structure and the shape of the molecule. Our other 3D visualization tool is Molecule 3D. So this has all of the capabilities of SPEC, including zoom, pan, and rotate. But in addition to that, it can support beautiful ribbon diagrams like you see on the right here. On top of that, you're also able to capture information about the user's interactions with the component. So if the user clicks on a specific atom or a residue or a chain on this molecule, you're able to enter that information somewhere else into your DASH app and use it to compute another value that will appear in the application. Among our custom chart types, we have clustergram. So this is a visualization that's typically used in gene expression studies to visualize hierarchical clustering of data, so showing which genes might be related to one another in their expression levels. So the cool thing about clustergram is that all the clustering analysis actually happens within the component itself. So all you would really need to do is supply your data, and then the component will calculate everything for you and display it in this beautiful visualization here. But of course, if you're a little bit more particular, clustergram also supports you defining your own functions that will be used to compute the clustering. And since clustergram has a plotly.py backend, you're able to capture user interactions like hovering and clicking in order to use those in another part of your app. So Circos is a really cool chart. It's a circular representation of data. So here we have the human karyotype, where chromosomes are separated by spaces and there are little bands on the chromosome that signify genes. You'll also notice these little red lines here, and those represent relationships between pairs of genes. They're called chords. So you can see how a chart like this might be super useful if you want to quickly, just at a glance, get a view of genes that are related to one another. In addition to this layout, Circos also represents heat maps, scatter plots, and histograms. And again, all of these will be in the circular layout. On top of that, you're able to capture, again, information about click data and hover data that you can use to feed into another part of your app. Ideogram is a way to visualize chromosomes. So these long strips here represent the chromosome, and again, the horizontal bands represent specific genes or regions on the chromosome that you would like to highlight. So right here, what we're seeing is the homology feature. So that connects one portion of one chromosome to a different portion on another chromosome. But in addition to this, ideogram supports adding annotations to give the user more information about what exactly they're looking at, and histograms. So for example, if you want to show the expression levels of a gene on the chromosome, you would be able to do that. And just like Sarcos and everything else that I've mentioned, you're able to capture click data and hover data. Sequence Viewer is a component that does basically what its name says. But the cool thing about Sequence Viewer is that it has this regex search here. So if you type in a regex string, the component will return back to you all of the matches, all of the things that match that pattern. And it will also return to you the locations of those specific matches. So you can imagine where you would be able to use that in another part of your application. In addition, you're able to highlight specific subsequences on the sequence and then define some behavior that will occur when a user clicks on it. And finally, you're able to capture information about the user's mouse selection. So if the user selects a substring with their mouse, you're able to, for example, give the amino acid count or give the molecular weight of what they've selected. So while we were developing all of these Dash-Bio components, we ran into a little problem because we were also developing demo applications in order to fully showcase their capabilities. But while we were doing so, we noticed that as we were visiting databases that are used in bioinformatics and downloading the data, we had to really work on reshaping it so that everything would plug into the components in the right places. So being true programmers, we wanted to factor out all of that hard work. So that's what we did with the Dash-Bio utils library. So this library contains a set of parsers for data files. And currently, it can handle files like soft files for gene expression data, FASTA files for proteomic or genomic sequences, PDB files for molecular structural data, among others. So the reasons that we wanted to develop this is because it makes our app development process a lot faster, and it makes our app code a lot shorter, and it really allows us to write only the code that we want to write. Instead of writing code that would just be wrangling data. Another reason is that we can allow for a direct database to visualization workflow, and we're also able to leverage the Dash app interface. So what I mean by that is that a researcher can literally just visit a database, download the data file that he wishes to or she wishes to visualize, and then go to the Dash app, upload that particular data set, and be able to interact with it and learn more about it without ever having to see the code that actually went into the app. So I'll do a little demonstration of that right now. So say that we visit this really interesting molecule of the month, actinomycin, and we want to see the structure using our Molecule 3D Viewer app. So what we can do is visit the PDB entry right here. This is the protein data bank, and it contains information about the molecular structures of biomolecules. So let's say that we want to download this particular structure. All we have to do is download biological assembly 1, and we have that file here. So then we can go to our Molecule 3D app. So this is one of the demo apps that I mentioned. It has the molecule. You're able to do everything I mentioned, like click on it. But that's not what we're here to do. We're here to see a different molecule. So all we do is we click on this upload component that's part of the Dash Core Components library. We can go into our file, select it, and we're able to view it immediately. And again, like I mentioned, it has all the capabilities of the Molecule 3D Viewer, like selection. You're able to view data about what you've selected. You can choose different ways to represent the molecule, and you're able to color by different parameters as well. Okay, so let's say that you just want not something this complex, but just like a quick and dirty type visualization. So this is a FASTA file. This contains proteomic sequences that are the output of a multiple sequence alignment tool. So something like Clustal-O would have used this, would have produced a file like this in order to highlight what a sequence alignment might look like. So this is a lot of data. Say that we want to visualize the consensus sequence or the conservation bar plot or the gap bar plot. So one way to do that would be in a Dash app. So just like I mentioned, all you have to do is open up your application. And this is what it would look like. So this is only 17 lines of code. And like I mentioned, I've defined the layout here, and I'm using our alignment chart component. So just within these 17 lines of code, you'll see that we're able to have a really rich and informative visualization. So let me show you what I mean. All you have to do to run this code is run the Python file that contains it. And then you'll see that it's running in my browser on localhost. So let's visit that. And this is what those 17 lines of code can produce. So you'll see, just like I mentioned, the conservation bar plot, the consensus sequence down here, the gap bar plot, and also you're able to navigate to a specific part of the sequence with this range slider down here. Okay, so that was pretty simple. Let's say that we want to analyze something a bit more complex. So this is a soft file. So like I mentioned, these are used in gene expression data experiments. The top of it contains some metadata about the experiment. And then right underneath, we have all of the numerical data related to the gene expression levels. So say that we want to perform a hierarchical clustering analysis on this. And we also want to extract the relevant metadata with our -bioutils library. So, and then say that we want to display the clustering analysis using the clustergram component. And on top of that, say that we also want to determine which rows and which columns are used to compute the clustering from within the app. So an app that does that would look like this. So you can see this is quite a bit bigger than the previous app that I showed you, but it's still fewer than 60 lines. So here I've defined the layout. So what I have is two drop-downs, one for the selected rows and one for the selected columns. Underneath I have just a little container that will have all of the metadata that I mentioned earlier. And then right underneath we have, of course, our graph. And then for the logic of the application, we want the graph to update, recompute the clustering, and re-render every time we change one of our row selections or our column selections. So let's see what that app looks like. And that app is going to look something like this. So we have, like I mentioned, the drop-downs up here. On the left, we have all of the metadata in a human-readable format. And here we have the actual clustergram. So, and again, like I mentioned, you're able to, on the fly, from within the app, choose what gets used in the clustering. Okay, so both of these apps, they're still pretty simple. So I wanted to show you something that really showcases the capabilities of Dash and how powerful it can be. So this is the app that I mentioned earlier that was used in that bioinformatics research publication. So you can imagine how a scientist might be able to use this in order to, for example, put in their own DNA sequence that they would wish to cut, put in a custom cut site, and then scrolling down, you're able to view all of these statistics about what will happen if you do cut that strand of DNA there. So something like this, you can imagine being useful to many, many people. And again, like I mentioned, this is running in my browser. It's a website. So any researcher who has this particular problem is able to visit this website and get a thorough analysis of their data. Okay. So since everything, again, that I want to reiterate, everything that I just showed you was free, open source, and MIT licensed. So you might be So is Plotly free? So yeah, everything that I just showed you is free. But our open source development is actually largely funded by companies that wish to see a specific set of features or components to enhance Dash applications that their teams have already created. In addition to that, we also partner with companies to create customized applications that meet the needs of their data science teams. And on top of that, we also offer Dash Enterprise, which is a suite of features for companies that want to level up their Dash workflows by creating enterprise-grade applications that have additional features like authentication, user analytics, pipeline reporting, and built-in designing. So many of these features were actually developed by us while we were working on applications for our commercial users. And they made our own internal workflows a lot smoother. So we thought that it would be a good idea to make them available to data science teams that really need them. So I want to thank you for your time and attention. These are some resources if you want to learn more about Dash Bio or about Plotly. And I'm happy to take any questions that you have at this time. Yep.
So what's the preferred method for launching web app, and is it scalable for heavier traffic?
Okay, so the question is, what's the preferred method for launching the web app, and is it scalable for higher traffic? So I'm not sure what exactly you mean by launching the web app.
The website said Flask.
Yeah, exactly. So Dash runs on a Flask backend. So Dash app is essentially just a Flask app. And as for high availability across users, that is a product that we offer. But of course, if you have your own server and you're ready to handle that kind of traffic, you're able to run a Dash app on your own server as well. Oh, yep.
Yeah, we bioinformaticians generally analyze data and deliver something to the PI, the doctors.
Mm-hmm.
I mean, how the doctor access the apps and stuff. I mean, we can post apps for everyone, for every data set we analyze.
Yeah, so the question is basically how are you able to share these applications with the people who might want to look at them? Okay, so like I mentioned, within Dash Enterprise we have a feature Dash Snapshots that allows for built-in reporting. So you're able to capture the state of the application after you've interacted with it in specific ways. And then something like that you would be able to present to your PI. But in addition to that, you're able to create reports with Dash if you want to. The interactivity is solely up to you, so you don't necessarily need to include that. You can hard code everything in.
The report will be static, small animations and actions and stuff.
Yeah, the report can be static, but it can also be dynamic, like the applications that I showed you.
In order for the report to be dynamic, there has to be some server to run the report, right?
Exactly, yeah. So you can use your own server, like I mentioned. You can use a server that we support and create, or you can use something like Heroku that hosts web apps online, and you can use that for your deployment.
That's generally not possible for the doctors to access that.
Okay, yeah, I'd love to talk to you more about it after. Yep.
Yes, we started using Dash quite a bit in molecular sciences as well. I was curious, one of the issues that we have with it is actually integrating it into pre-existing websites, because Dash seems to like to take over the entire table. Do you guys have any thoughts about the future of that and being able to integrate it?
Okay, so the question was, do we have any plans for Dash to be able to be integrated into websites? So right now, unfortunately not. The way that you're able to put in Dash apps is, I would say, through using something like an iframe and hosting the application somewhere else. But right now, we have some work done on Dash Embedded, but that is, once again, one of our enterprise products. Yep.
Great demos.
Thank you.
I was wondering about, like, for example, the RNA-seq or proteomics data. Like, these files can get pretty huge, and our researchers sometimes just don't have enough disk space on their laptops to be able to, like, download and then upload these data. So I was wondering, have you guys ever worked with like S3 buckets or use AWS CLI or something like that to load the data? And is there some kind of like, do you guys have some utils for that to make that more seamless?
Okay, so the question was essentially, what do you do if your data files are huge and you don't want to download and upload them all the time? So in the demos that I showed you, we're not using any cloud services, but we do have a lot of users who are able to integrate that into their Dash applications in order to serve large data files, like you mentioned. Yep.
Do you guys have any plans of porting some of these components from the Python and R APIs to the JavaScript APIs?
So yeah, the question was, are we porting some of the components from Python and R to JavaScript? So the cool thing about Dash Bio is that a lot of the components, especially the ones that I showed you, are based on existing React JavaScript libraries. So all we did was wrap those components to be compatible with Dash. So you're able to use those open source libraries in JavaScript if you want.
So what did you use for the molecule viewer?
The molecule viewer is Molecule 3D for React. It's a project by Autodesk.
Okay.
Yeah. Yep.
Do you provide any of these as Docker containers? Or maybe your users have made Docker files and whatnot?
Okay, so the question is, do you provide any of these as Docker containers? Not what I've showed you today, but I do know that some of our users use Docker. You can visit our community forum. I'm sure there are lots of threads on it, but unfortunately I'm not too familiar with that. Yep.
We have all Flask-based the patient color we incorporate with those route components.
So you're essentially asking how would you incorporate Dash into a Flask application? So Dash is already running on a Flask backend. So if you have like a more detailed use case, I'd love to talk to you about it after this. Yeah. Yep.
Your soft parser, you showed that it parses the platform metadata and the expression data. Do you actually parse out the sample level metadata as well?
Okay, so the question is, for the soft file parser, do we actually lose the data about subsets of the...
Yeah, there's some soft files will submit actual metadata on the samples themselves.
Yeah, metadata on the samples. So yeah, we do. We just didn't use that in this particular application. Yeah. I think we're good. Oh, yeah, sorry.
I already asked a question.
That's okay.
So Dash works on Flask. I was wondering, has anyone tried using Dash with Tornado or other Python frameworks?
I'm currently not aware of that. But again, like I mentioned earlier, you can visit the community forum, and I'm sure there's somebody who's asked that question too.
Okay, let's play your speaker one more time.